Thank you, Barbara. Thank you for a nice introduction, and thank you for the invitation to be here. I've really enjoyed talking with lots of people today. It's been a really fun day. I just slightly disappointed that I couldn't actually be in person, but this is 2020. So yes, my title, sort of fairly hyperbolically, is Inferring the Ancestry of Everyone. And I'm not going to infer the ancestry of everyone, but I am hoping to convince you that at least this is something that may be possible someday, and B, that this is something that we would actually want to do. So the context that I'm sort of thinking about is where we are today in 2020 is that we're collecting genetic variation data at unprecedented and ever increasing rate. So we have in the human genetics world, we have things like UK Biobank, which is 500,000 samples that are genotyped. We're going to have something like 100,000 whole genome sequences within the next 6 months, and there's plans to sequence another 100,000 more. After that, we've got Nomads, which is some number of 100,000 of genotypes and sequenced genomes. And we also have things like the Darwin Tree of Life, which is trying to sequence whole genomes for 60,000 eukaryotic species in Britain and Ireland, if I remember correctly. So I think it's fair to say that we're now in the era of what I call mega sample genomics, where we think about the number of samples that we're dealing with in the millions, and this is becoming an increasingly standard thing to do. And this is fantastic. We're learning things about the world and about history and demography and medicine from this. There's huge insights being gained into the biology. But from a computational perspective, we've got some very serious problems sort of facing us. And this is because the methods that we're sort of working with today predominantly are things that we developed largely in the era of the 1,000 Genomes project. So things like the variant call format, these developed out of projects like 1,000 Genomes and are appropriate for things that are measured in the kilo sample scale, and we have thousands of samples. But when we have millions of samples, then these methods start to break down pretty badly. And the reason that they break down is that the model that we have for representing and thinking about the data is fundamentally one that doesn't scale. So the defining structure of kilosample genomics for me is what I call the variant matrix. And a variant matrix, for those of you who are familiar with it, is literally a VCF file. So we've got a matrix in which the columns correspond to our sample genomes, and the rows are positions in these genomes at which variation occurs within our sample. And we literally just write down the observations for our samples at these positions. And we assume that for positions that aren't samples, they're equal to the reference genome. So we can say that usually our number of variant sites that we have is something like 1 to 100 million. And then clearly the complexity of storing and working with this data structure is order number of sites times number of samples. It's sort of unavoidable quadratic complexity here. And when the number of sites is 100 million and the number of samples is 1 million, then this is a very, very large and cumbersome matrix. And it's also a highly redundant matrix. There's a lot of structure in there that we're not capturing by just writing it down in this very simplistic way. So I want to advocate for a different way of looking at the data. And it's a very old way of looking at the data. It's what we've been doing for decades, or maybe even a century or more. And that we use the concept of a tree to encode the ancestral relationships within our data. So we're all familiar with these sort of phylogenies. It's how we relate a bunch of different species to each other. So, for example, we've got the phylogeny of the humans, chimps, gorillas, and orangutans, which I hope I've got in the right order here. And it's a very intensely studied problem. We've got probably hundreds of different ways of inferring phylogenetic trees. So from the context, or from the perspective of using and working with our data from a sort of computational perspective, trees are extremely powerful because it changes our fundamental complexity from being linear in the number of samples to being constant. So if we know what the underlying tree for a given site is, then we can encode the observations for that site in a constant amount of space. So assuming that there's a small number of mutations, which there nearly always is, then we can encode the observations, so the A's and T's that we see here at the leaves, we can encode them in 3 bits of information. And when there's a million leaves, then this is quite a large difference. Trees are also powerful from a conceptual perspective. So we, in phylogenetics, the first thing that we do really is to estimate a tree, and then we base all of our subsequent analyses on that tree. And so if we knew the trees behind our data, then most of the problems that we have in evolutionary biology would be fundamentally easier. And the reason for this is that the structures that we use today, like the varying matrix, like the site frequency spectra and PCs, they're all just low-dimensional summaries of trees. There's information is lost when we project down from the trees onto the observed sequences that we have today. Then huge amounts of information are lost, and PCs and site frequency spectra really are a very crude sort of summary of that true history. So trees are actually, there were trees that underlie our data. They did happen, and they capture all possible observable signals that we could hope to have in the data. So if we can estimate trees accurately, then we can ask questions about these trees and hopefully have a much richer data structure to base our analyses on. And finally, the sort of background material, of sort of motivational material, and I guess it's sort of building on the last point, is that when we actually want to infer things about the evolutionary past, often how we structure this is to formulate some sort of hypothesis on the tree in terms of the genealogies that would have happened. So, for example, we might say that if we wanted to detect selection, we might think that selection makes trees unbalanced, giving shorter branches under a selected site or under a selected mutation. And then what we do is we translate this prediction, which is directly about the trees themselves, and we take this prediction and sort of map it back down onto the space of genome sequences again. And so we're taking some prediction that we have about the genealogies, and we're making it less precise by pushing it down onto the sequence data, because the observable effects of having a particular pattern of short branches in your trees, well, that could happen in a number of different ways. The things that we observe about the data in terms of the site frequency spectra or PCs or whatever, then that could have happened in a number of different ways. So there's sort of level of precision lost by translating our predictions back into the space of sequences. And Kelleher makes this point really well in a review last year in Nature Genetics. So if trees are so great, then why aren't we using trees for everything already? And the answer is, well, we can't, or at least we couldn't until pretty recently. And the reason that we can't is that recombination means that we don't have just a single tree, as I'm sure most of you are very well aware. We don't have a single tree along the genome. We have a whole bunch of correlated ancestral histories. So basically what recombination does is it takes two parent sequences and it merges them together to make a single child sequence. And the upshot of this is that the ancestry of this child sequence is different on the left-hand side of the recombination breakpoint to what it is on the right-hand side of the recombination breakpoint. And when we play this out over evolutionary history, over evolutionary time, what this ends up giving us is this sequence of different but highly correlated trees. So I'll return to this again in a bit, but basically what we have is a set of trees which relate the same set of samples to each other along our genome of length 100 here. And these trees are clearly very highly correlated. They're not, totally distinct trees. They have shared shared branches and shared nodes. And we want to capture this correlation structure if we want to understand what the actual history of our samples was. So this is something that we've sort of wanted to do for a long time, is to infer the ancestry in the presence of recombination. But unfortunately, it's really a very difficult problem, and it's usually referred to as a problem of inferring ancestral recombination graphs, or ARGs. So we've been thinking about how to infer ARGs for about 3 decades, and because of the sort of combinatorial structure of ARGs and the general approach that we were taking, it's a very, very difficult thing to do with statistical rigor. The space, the space of binary trees alone is enormous. When you take into account all the different ways that you can have binary trees along a genome and interrelating to each other, then the combinatorics blows up extraordinarily quickly. So it's almost impossible to do ARG inference in a sort of rigorous statistical way that doesn't make an awful lot of approximations. So basically, until recently, the methods that we had for inferring ARGs only really worked on toy data sets, so literally a handful of samples across tens of sites, and it wasn't really applicable to the data that we actually have and what we work with. So I want to convince you that the succinct tree sequence is a data structure that can solve these problems that we have of profoundly increasing scale of data, of capturing the richness of biological signal along the genome and providing a rich substrate for which we can base our inferences on, and also give us a rich software ecosystem using some shared infrastructure. So the succinct tree sequence is what this talk is going to all be about. And as I say, it's a new method for capturing the correlation structure of trees along the genome. And technically, it's actually a more general structure than an ARG, which is why I kind of avoid calling it an ARG. But for those of you who are ARG aficionados, you can think of it as an ARG. And I'm also going to be talking about the software that we develop as a sort of key part of that in this core TS-Kit library. So I'm not going to talk too much about simulation today. I'm going to focus instead on the problem of inferring ancestry. But I think I should really give some sort of quick overview of what we've done with this data structure in the context of simulations. And so over the last 4 or 5 years, I think it's fair to say that there's been a real step change in what we can do in population genetic simulations, and tree sequences have been a core part of that. So coalescence simulations are now, we're able to run them at a scale that would have been unimaginable previously. We can simulate whole genome ancestry under the exact coalescent model for millions of whole chromosomes. And this is something that we routinely do now using software MS Prime. This is the simulator, and we can analyze the output of those simulations using TS-kit. There's also forwards time simulations. So there's a sort of follow-up piece of work that we performed where we take the ideas of using this data structure, which allows us to record whole genome ancestry very efficiently, and we apply that to the problem of forwards time simulations. And the upshot there is that we can often simulate interesting models several orders of magnitude more quickly than we could before, and we can also retain far more information about the actual simulation. So not just running a simulation and then printing out a few sequences at the end and sort of trying to infer what happens within the simulation based on those sequences. We give a complete record of every parent-child relationship in the history of the simulation ever. And all of this is encoded extremely compactly, and often much more compactly than just outputting a small subset of the sample sequences. From a software perspective, I'm really pleased that these simulators all actually interoperate with each other now. So we can do things like run a coalescent simulation to make some neutral burn-in, and we can put that, we can use that as the initial condition for a forward simulation, and all of the ancestry will be retained, and all of the from the end user's perspective, it all just works. You provide a TS-kit tree sequence, and SLIM or forward pi11 will take this, simulate some stuff on top of it, and give you back a tree sequence, and you'll have the full history of everything that happened. And also, just to briefly plug the standard PopSim project, which is the work of the PopSim consortium. We've also standardized there on using TS-kit tree sequence as the output of that simulation package. So there's a number of, if you want to catch up on the state of play with simulation using tree sequence technology, I'd recommend these papers here. And if you're not using MS Prime or SLIM or forward pi11 for your simulations, I really highly recommend giving it a look, because we can genuinely do things now that were just plain impossible before. So that's simulations. I'm not going to say any more about them, and I'm just going to quickly explain what this data structure actually is and how it works. And it's very simple. There's really no trick to it. All we're doing is writing down the ancestral relationships between nodes in a simple sort of numerical format. So on the left here, we've got two different tree topologies, and they apply to two halves of our genome of length 10. So left to right here is our long genome. And for example, we can see that this edge between child 1 and parent 3 is the same in both of these trees. So traditionally, if we were going to encode these trees, we would write down a Nuix string, and this edge would be written down explicitly in both of those trees, and that's a waste of space. Now, we know that this edge is the same. We know that these nodes are the same, so we really only need to write it down once. And we do this. We say, from position 0 on the left to position 10 on the right, the child is node 1 and the parent is node 3. And that is literally all there is to the encoding. This way of doing things captures the correlation structure among trees, and it's very compact, as we'll see in a minute. So to sort of make this point, I did some simulations where we simulate a human-like chromosome, so 100 megabases of human-like sequence. So it's sort of intermediate-sized human chromosome. And we do that from 100 samples up to 10 million samples. And on the y-axis then is the size of the file needed to store this information on a log scale. So this is a log scale. And what we see then is that to store all of the ancestral history and all of the variation data for this simulation of 10 million 100 megabase human chromosomes, it only takes 1.2 gigabytes of space. And if we were to do that in the standard VCF form, and that's just to write out the sequences, there's no ancestral history recorded here, it would take about 13 terabytes. So there's a 3 order of magnitude difference in the amount of space that we need to store this simulation data. And if we sort of extrapolate this out to another few orders of magnitude, where we think about 10 billion samples, it comes out to something like a terabyte of space you'd need to store a tree sequence of 10 billion human chromosomes. And I forget what the amount of VCF is, but it's academic. But the point is that it becomes feasible, in principle at least, to store the history, the complete genetic history of literally every human being alive, to store this on your laptop and process this on your laptop, which is where our title comes from. So it's not possible to do things to infer the ancestry of everyone at the moment, but it is possible in principle at least someday.
Sean, can I interrupt you for just a quick second?
Of course, yes, please.
Sorry, Magnus here. Sorry, I'll just make myself a bit more impersonal.
So,
right. I mean, you're talking about what you're really saying here is that under certain assumptions, it's possible to simulate, right? I mean, so if you could infer, you could in principle store it, right? I mean, but that's, I guess you're not really making a bigger claims than that. I mean, that's a very big step from actually being able to infer this, right? So if God gave you the answer, you'd be able to store it, right?
I completely agree with you. If God wrote down the actual ancestry of everybody, that's how we do it.
All right. Okay. Just wanted to make sure that we're sort of in the.
Yeah. I'm being deliberately provocative.
Yep.
There's clearly we couldn't actually do this right now, but I think we've made a number of order of magnitude steps towards it. So as we'll see a bit later. So I don't think it's completely.
When you simulate this, so you're making this kind of Markovian approximation, or
this is the full coalescent recombination. This is,
wow.
So that's for 10 million human samples. It's actually faster for us to simulate the full coalescent than the SMC.
Sorry, Jerome, it's Nick here. I'm just interrupting you. Isn't it the case that even if you can't infer the actual ancestry of everyone, you could infer an ancestry consistent with the sequence data and use this simply as a way to compress the sequence data? Is that a useful thing to do?
Certainly, yeah. And this is very much what I would like to do, is to use this technology as a way of compressing genome sequence.
Yeah.
And it's inferring the actual history of anything is quite a tall order. At best, you're going to infer something which is consistent and has some sort of plausibility about it. And I think that's what we're hoping to do.
Mm-hmm.
Is there anyone else who wants to jump in at this point? I'm very happy to answer questions as we go along. No? Okay, I'll keep going. So normally when you have something that's very highly compressed, you have to pay a price of decompressing it before you can actually do anything with it. But it turns out that this is the opposite when we store genetic data using the underlying ancestry, the underlying trees, then actually computing the things that we're interested in becomes much faster, because all of the things that we actually want to compute, things like population genetic statistics, they're actually defined in terms of the underlying trees in the first place. So we can sort of short circuit this tedious round trip that we go through this big matrix and compute things from that, and instead we just look directly at the trees. And when we play this out, as with these simulations, so again we've done some simulations on a log scale from 100 samples up to 1 million samples, and when we want to compute the genus D from all of these, if we use sort of these well-regarded libraries that are based on the variant matrix, you can maybe get to 300, 400 variants per second when you're doing this. But if we use TSKit, directly on the trees, we're looking at 200,000 variants per second. So we get a 3 orders of magnitude speed increase by working with a version of the data which is vastly compressed over if you want to see it as the source matrix data. So there's really compelling case here for if we could encode our data as trees, then we'd burn a lot less electricity and we'd spend a lot less money on storage, even from these very mundane sort of boring perspectives. So that's the computer science motivation for wanting to estimate trees from real data. We want to be able to compress the data, and we want to be able to apply these very fast algorithms that we can define directly on the structure. But of course, much more interestingly to this group, the history itself is of great interest, and if we can estimate something which is reasonably plausible and consistent with the data that we observe, then this could be very useful to us in the questions that we want to ask in biology. So I'm going to give you a quick overview of how our algorithm for inferring this ancestry works. I'm not going to get into the sort of gory details. If you're interested there's a paper from last year, in 2019, and there's also a really good philo seminar from Yan Wang up on YouTube, and there's some links to that later on in the talk as well. So TS-Infer is what the method is called, and what it does is it takes some input sample data. So our input data is a set of phased haplotypes in which the sites have all been polarized. So we know that we know what the ancestral state is, and we encode that as 0, and we know what the derived state is, and that's 1. And we're only dealing with biallelic SNPs at the moment, but this can be sort of extended in a reasonably clear way. So what we do is, as we progress through the algorithm, we're inferring haplotypes for our ancestors, the genetic ancestors of our sample. And as we go through time, we'll fill out these haplotypes, and we'll also, on the other side over here, build some trees as we see how these haplotypes relate to each other. So the first thing we do is we have a most ancestral haplotype, and that's the ancestral state everywhere, because that's the ultimate ancestor of everybody. And then we create an ancestral haplotype. Actually, that's also the most ancestral. So we come to a more interesting ancestor, and this is an inferred ancestor. So this is the ancestral haplotype that we think the mutation for this site arose on. So this site is a tripleton, which is as frequent as something can get in this sample. So we guess that this is among the oldest mutations. So basically, the haplotype that this arose on is 0 everywhere else, is the ancestral state, and it's 1 at the focal site. We do the same thing for this site over here. We've got some heuristics for figuring out what these ancestral haplotypes are, taking into account recombination. Taking into account recombination, then we figure out how these haplotypes relate to each other. So I've been sort of skipping the part over here where we show how they relate to each other. So what we do is, once we've inferred an ancestral haplotype for a given site, we then see how this relates to all of the older ancestral haplotypes by using the Lee and Stevens copying process. So the Lee and Stevens copying process here has decided that this path through ancestor 3 and ancestor 2 is the most likely explanation of this this haplotype that we have generated. And this is reflected over here in our parent matrix. So we see that the parent of 4 is 3 for all of these sites, and then it's 2 for the rest of the sites to the left of that. And this is a familiar sort of operation of recombination. And the outcome of that then is that we have not 1 tree, but we have 2 trees. So these trees share the same structure, except for how 4, the ancestry of 4. So on the left, the parent of 4 is 2, and on the right, the parent of 4 is 3. And once we have generated these ancestral haplotypes and we've figured out how they all relate to each other, we then go through our sample haplotypes and see how they relate to the ancestral haplotypes. So we go through all of our sample haplotypes, and when we found these copying paths, we have now got these complete histories. We've got complete trees relating all of the samples to each other, and we've inferred an ancestry for the data. So this is a heuristic process. So it's heuristic in that we use heuristics to generate the ancestral haplotypes, and we use this approximation of this very sort of broad brush approximation of coalescent dynamics from the Lee and Stevens process, but it seems to capture the essential dynamics pretty well. So I'm just going to show a quick comparison of TS-infer with some sort of existing methods, Argweaver being the most interesting one. Argweaver is sort of the gold standard in terms of accuracy at the moment. So basically what we see is that TS-infer and Argweaver are more or less as accurate as each other. So we're varying the mutation rate across the x-axis, and what we're showing on the y-axis then is the average tree distance between the original trees, the trees that we simulate, and the trees that we infer. We calculate the distance between these trees using the KC distance metric, and we take an average over the output. And then what we see is that as the mutation rate increases, as we get more and more information, and this is just infinite sites mutations, there's no recurrent mutation here. As we get more and more information, the distance goes down. So all of the methods become more accurate. And when we put in some error, then we see that Argweaver is a bit more accurate than TS-infer, but it's not. There's not a massive difference. Where we do see a massive difference then is in the running speed. So at the scale that we can actually run Argweaver, this is the sort of time difference. Argweaver takes 2.5 hours, whereas TS-infer takes some small fraction of an hour. I can't remember what that actually is. But we're limited to using really, really tiny samples here. 20 is really the limit of what you can actually work with in Argweaver. And this is a very small chunk of sequence as well. It's only 200 KB. And when we scale things up to real sort of samples and real chromosome lengths, then we see that TS-infer scales really remarkably well. So when we've got 10 megabases of human-like sequence for a sample size 10,000, it's taking a half an hour to do this whole inference, which is really quite remarkable, the difference in scaling here. And so, this is approximately linear with the growth, so we can see, we can extend our sample size out from 200,000 up to 1 million, and maybe someday 10 million, who knows. So I want to show some results just to try and convince you that the things that we're inferring have some biological signal. It's not just a sort of encoding of the data that's useful for compression and computing. We're capturing real biology here, and so I'm going to show this genealogical nearest neighbors metric, which is a measure of the topology of different trees. And I'm not going to go into much detail about what that is. I'm just going to try and show you the high level stuff. So the first thing I want to show is this is the Peruvian 1,000 genomes population. And what we're showing here is the sort of a measure of the relatedness between an individual in the Peruvian population and the other 1,000 genomes continental groupings. So the people on the left have more European ancestry, and the people on the right have more sort of American ancestry. And we picked out this one individual here as being really interesting because they're clearly very recently admixed. So we've got an individual here who's got a lot of African ancestry, a lot of Asian ancestry, and a lot of European ancestry. And you can see the recombination breakpoints. There's clearly a recombination that's happened in this area. And then their other haplotype, their paternal haplotype, say, well, that's much more sort of standard American. And we can see that there's a bit of noise here. This isn't perfect inference, but it's capturing important biological signal. And then when we sort of scale up a bit from here, we think about taking this per person sort of measure and averaging that out over the whole population. We can then compare pairs of these human populations across the 1,000 genomes sampling groups again. So we do some clustering on this. This is a standard sort of cluster map plot from Python. And nicely what we see is that all of the African groups, they all sort of cluster together as we might expect. The Europeans all cluster together, and they have this sort of complex relationship structure with the Americas, very much as we'd expect. So I think it's fair to say that we're capturing some real biological structure here. To convince you that we can scale to the really large scale data sets, I'm going to show you some results from UK Biobank as well. So we inferred trees for the 500,000 diploid samples from chromosome 20. This is genotype data, so it's quite sparse, and that we had to use some extra methods to improve our inference on that. And it ran in a couple of days. 500,000 human chromosomes, we can infer ancestry of that, and we get back a file which is perfectly workable. And one of the things we did with this then was do a similar sort of relatedness structure plot on the people in UK Biobank. And again, this tells us things that we would expect to be true. So we see that people from, say, East Wales are more likely to have genetic nearest neighbours that are from West Wales than sort of from anywhere else. And the people who are in Scotland, so people in Northern Ireland are more likely to have genetic neighbours that are in Scotland than rest of the other parts of the UK. So again, this is hopefully convincing you that there is real biological signal in the trees that we infer. So this has all been about TS-Infer, which was sort of published last year, and the methods. I'm sure some of you are familiar with this method and with these plots, and have been quite bored up to this point. So I want to give a sneak preview of some developments that are coming out pretty soon. So we're hoping to get a preprint out in the next sort of week, talking about some major developments that we've made more recently. So one of the big limitations with the current version of TS-Infer is that it only does exact matching. It only allows you to exactly match your target haplotypes against the reference panel of the ancestries. And while this gave us plausible signal, and it was a good stepping stone to allow us to actually scale first, is clearly something which we need to refine for real biological data. And the new version of TS-Infer is solving this problem pretty well by incorporating the full Lee and Stevens model, so that we now have variable recombination. We can sort of condition on a recombination map to say where recombination breakpoints are more likely to happen, and we can also have a mismatch term which gives us the relative likelihood of recurrent mutations. So do we prefer to have another mutation or to have a recombination in order to explain the input haplotype? And TS-Infer fully supports this now. We can have sort of arbitrary patterns of recurrent mutations to explain biology and sequencing error. It also supports missing data, pretty importantly, I think. So we're able to actually impute data using the ancestral haplotypes as a reference panel, and arguably this is a much more principled way of doing imputation than imputing against some panel of contemporary genomes. So I want to give just a quick results to try and convince you that this is something useful. So this is unpublished material, so please don't post this image on Twitter because we're still checking the details. But what this is showing is the number of mutations that we infer per site as we've inferred the trees, and as we're matching these haplotypes, we're inferring mutations based on these recombination likelihoods versus mutation likelihoods. And what we see is that most sites are explained by one or very few mutations, which is exactly what we'd expect. And this is reassuring that we're mostly seeing, most sites are well behaved, and they don't have a large number of mutations. But we do see some sites where there's really pretty, sort of ridiculous numbers of mutations, really, 400 or 500. And we wanted to sort of get to the bottom of why these were happening and what these were from. And so we looked at sites in the genome that have signals of being low quality. So one signal of low quality is being in the 1,000 genomes accessibility mask. So these red sites have all been marked as suspicious by the 1,000 genomes pipeline. And what we see is we have a highly elevated rate of large numbers of mutations among these low quality sites. And another signal of dodgy data are sites where there's basically no LD between them and adjacent sites. So the orange sites here are those in the genome where there's no LD at all between the current site and those surrounded. And we see those are also almost entirely, they almost all have very large numbers of mutations. So this seems like we're picking up signals of error in the data, and we're potentially able to correct that, and that's really exciting for me. We can find places where there are error, and potentially we can correct it by either throwing those sites out that have suspiciously large numbers of mutations, or we could try to correct it by putting some model on top of it, which, assuming the tree is true, and some model of where the errors came from. Another really important recent development, which we'll be talking about in the preprint, is our dating algorithm. So this is a method of taking the topologies that TS-infer produces. So TS-infer only produces tree topologies. It doesn't think about the times at which nodes happen at all. So it gives us no estimate of when particular variants arose, other than a partial ordering of them. But Wilder and Jan and Gill have developed a method called TS-date, which takes a tree sequence that's output by TS-infer. It uses some sort of coalescence reasoning to infer node times for these on this tree sequence, which then opens up a whole bunch of new applications in biology, because then we can really start asking questions about when mutations arose and, you know, how quickly they swept to fixation and all sorts of stuff. So this is, I think, really opening up the door to a lot of biological applications. And this is just a quick result showing you the sort of thing that we can do. So this is showing the pairwise time to the most recent common ancestor among the population groupings in the Human Genome Diversity Project. So there's a large number of different populations that have been sort of chosen to represent the genetic diversity of humans as well as possible. And we've taken this data and we've inferred a tree sequence using TS-infer, and we've dated the topologies using TS-date. And then we take the mean TMRCA between pairs of samples in these populations and then plotted them as a heat map. And again, clustered them using a cluster map. And the color then gives us the estimate of this time to most recent common ancestor. And, you know, we see sort of reassuringly biological things. You know, the African groupings, their TMRCA to basically everybody else is very old, and their TMRCA to other African populations is also very old. And this very much lines up with what we would expect beforehand. And we see things like the Americas, populations from the Americas, they have a very recent TMRCA, which again we would very much expect. So the details of these methods are going to be in a preprint, which we'll be hopefully putting out in a week or so. And so if you're interested, please keep an eye out and have a read. So yeah, I just want to finish up with a few thoughts, and I'm very happy to answer questions after that. So the perspective of software is really important, I think, in that, you know, we want to be able to work with these very large scale data sets, and we need well-engineered software to do this. And so we sort of envision the TS-kit library as being the central hub of all of that. So it's an open source library. It's up on GitHub. It has a whole bunch of methods for doing a lot of different things. And it's an open source community. We're always interested in...
Calcium is fine for Camillo.
I think someone's got their microphone on. And we're also always interested in finding new developers. So please do check us out on GitHub, and we have a Slack channel if you're interested in joining that. You know, there's a community, and we want to welcome new members. So I'd also like to think about the general approach here. Well, firstly, from a sort of computational perspective, and you know, given that we're sampling literally millions of human genomes now, and probably millions of other genomes as well when we put them all together, we can't really just, you know, throw money at Google and hope that we will be able to do the computations that we need. Right? Genomic data is highly specialized, and you can't just use existing methods on genomic data to and expect to get good results. We need specialized methods which are guided by population genetics theory, and which I think is where the tree sequence comes in. So it, to me, elegantly combines the computational efficiency that we need with the biological richness. So it's representing the information that we want, but it's also combining this fairly compelling efficiency of storage and processing. So I think there's a virtuous cycle there. As we understand the evolutionary forces that shaped our data, we can store and process it more efficiently. And then by having more efficient algorithms to interrogate the data, we understand the evolutionary forces better, and we can formulate different questions. And then hopefully we can, infer better trees, and then again store and process our data more efficiently. So yeah, if you're interested in more information, there's 2 really good Philo seminar videos on YouTube from Walter Wolms and Yan Wang, who've done a huge amount of the work that I've been talking about here. There's a paper from Nature Genetics last year. There's also the statistics paper, which gives a good overview of the actual data structure itself. We're on GitHub, so please check us out there. And as I say, there's also a preprint coming out in a few days. So please keep an eye out for that and have a read. And with that, I'll happily to answer any questions people might have.